the protocol you just talked about, what happens if the mutation that you make is not viable? Do you just not recover any genomes, or what happens there? Do they survive in mixed infections or something?
So in the small number of cases that we've looked at, if the gene is essential, the mutation appears and is identifiable within the primary plating. The reason is that primary plating is done as an infectious center assay. The cell has not been allowed to lyse prior to plating, simply by plating early. So, for example, we constructed a mutation in one of the lysine genes, which we believe is required for formation of a plaque because it's needed for lysis. The mutation appears in the primary plaque because it's growing in the presence of helper phage, wild-type helper phage. A simple way of determining whether a function is essential or not is as follows. You take that primary mixed plaque identified by PCR. There's typically 10 to the 6 to 10 to the 7 particles in a plaque. So you can dilute for single plaques, or you can simply plate on an agar plate where there's maybe 100, 200, 300 plaques. Quite a large number of plaques, but not enough to get a mixed infection. So you get 100 or 200 plaques on a plate. You simply flood the plate and collect the totality of the particles and just do a PCR reaction. If the gene is a non-essential gene, if the mutation is tolerated, you can clearly see the mutation in the primary plaque and in the secondary lysate. If it's in a gene that's required for plaque formation and growth, it is present in the primary mixed plaque, but it's absent from the secondary lysate. And in the cases that we've looked at, that's what we've discovered. Does that make sense?
This gets to the, you know, it's sort of a truism now. Oh, sorry. People will say, oh, 1% of microorganisms are culturable. So I'm curious, the issue of what percent of phage are plaquable, and is there something about the host we're using that promotes our getting as many as we can? You know, when you listed the things that prevent you from seeing something, restriction modification you didn't put in that list, but it's one that's there. And isn't there supposed to be some kind of siRNA type of thing going on now with respect to phages, and how are we going to deal with that?
Okay, so first of all, the only phages that you can identify and characterize are the ones that you can identify and then characterize, right? And yes, there's 10 to the 6, 10 to the 7 particles in per mil, or the equivalent of per mil in the solid samples that you have. Clearly, not all of them infect mycobacteriophages. It would be a lot easier if they did. But knowing what they infect is much, much harder. And it's harder for a couple of reasons. One, obviously, if you don't have the host, then if you use the typical isolation techniques, you don't find it. You could ask, well, there's lots of particles there. Let's just collect a whole bunch of them, and we'll do metagenomic studies. We'll take all these bacteriophages, we'll just sequence them at random. And what's more, that'll not only tell us about what's there, if there were signature sequences that indicated what host they infected, then it would begin to address the question. So people have done that. There's been quite an extensive amount of metagenomic studies on bacteriophages. Part of the problem is that the current picture shows that the diversity is very high, and so you just find a lot of genes, a lot of sequences that are just sequences that don't match anything. The diversity is high, so getting assembly of genomes is difficult. And from what we can tell, in most cases, there are not signature sequences that are a giveaway for which host is being infected by a particular bacteriophage. Now, so that's what part of that's the answer to one part of the question. The other part of the question is other reasons why you don't see plaques. Yes, I gave a short segment of what is actually a very long list. And
I wasn't looking for that, and maybe this stopped me when I'm getting inappropriate. I thought it surprised me.
Microphone.
I'm sorry. It surprised me that we are all using the same host, because I kind of imagined this thing as we're going to get a mycobacterium from our environment and get the phages that infect it. And I'm not saying change the protocol, but that's why I probably shouldn't be raising this question
So
now.
I think that this is a great point.
Good things for discussion
Because
elsewhere.
it's a
Yeah.
philosophical
Yeah.
discussion that can really go on.
Stop me.
So, and we have staff from Genetia Farm that are going to come in here and talk about the safety issues with respect to being in the lab here, and they're going to come in 10:15 minutes. So I would suggest that we stop, you guys take a very quick break. There's coffees right outside this room, and back in here by 10:15. And Graham is going to be around a while, so you can ask him some questions. Also, those of you who are taking notes, don't worry to get all the notes. We did video record them, and these lectures are going to be uploaded onto the Wiki, so you can download them anytime for teaching purposes, and all of the PowerPoints are going to be presented on the website. So you're okay. So let's take a quick break. Thanks again.
Thank you.